Hi, my name is Marissa Fuss, and this is my Advanced Concepts presentation 2. Okay, so the prompt. For this presentation, your goal is to walk the audience through how oxygen binds to heme, and if any structural changes occur, along with explaining the binding capacity of oxygen to heme in the lungs and how that differs from that in the capillaries. Also explain how CO2 interacting with water can affect the pH of the capillaries. Okay, so some background information. Heme is the prosthetic group that is the determining factor on whether myoglobin and hemoglobin are able to have oxygen attached. It is important to understand that heme is what allows the blood and muscle to have the distinctive red color that it does. In order to understand how oxygen binds to the heme group, it is essential to understand the structure of heme. The structure of heme will be discussed in the next slide of this presentation. Okay, so the structure of heme. Heme contains an iron atom, which is seen right here on the figure to the right. It also contains photoporin. Protoporin is considered to be the organic piece of the structure. It's comprised of 4 chiral rings that are connected through methane bridges to ultimately create the tetrachiral ring. So you see the chiral ring right here, and they're all around the structure. The iron atom connects the chiral rings through nitrogen atoms, which you can also see right here. In standard conditions, it's important to know that the iron is considered to be in oxidation state. At this time, the iron atom has the ability to create 2 additional bonds. These bonds can be on opposite sides or what are known as the heme plate. Okay, so structure of heme continued. These additional binding sites are referred to as the 5th and 6th coordination sites. On the 5th coordination site, it contains an imidazole ring of histidine residue of the protein. It is commonly referred to as the proximal histidine. On the other hand, the 6th coordination site is not used because that is where the oxygen is able to bind. The iron ion is located around 0.4 A under the polyporphyrin plane because of its size. It is important to know that there is a distal histidine present in the structure that is on the other side of the proximal histidine. The distal histidine is important because it does not allow the iron to be oxidized into ferric ion. If the oxidation did occur, oxygen would not be able to connect to the heme structure. It is also important because it decreases the chances of carbon monoxide to connect to the heme structure. Okay, so now oxygen binding to heme. The binding of oxygen to the heme structure cause a noticeable difference in the structure of the overall combined structure. So the electrons are reshuffled, and this causes the iron ion to reduce in size. So from the deoxyhemoglobin to oxyhemoglobin, you can see that the iron significantly reduces in size and is able to move into the plane. Okay, so more specifically, there's a hydrogen bond that is created between the oxygen atom and the distal histidine of the heme structure, which you can see to the right. Okay, so how oxygen cooperatively binds to hemoglobin. Hemoglobin has a quaternary structure, which you can see in the figure to the right. Hemoglobin A in humans contains 4 subunits, which contain 2 alpha subunits and 2 beta subunits. This arrangement of subunits is similar in myoglobin. More specifically, hemoglobin's 3D structure can be described as one pair of alpha-beta dimers. These dimers are the same to each other. These dimers are what ultimately give hemoglobin the tetramer shape. When hemoglobin is deoxygenated, the alpha-beta dimers are connected in a specific manner, and one of these locations are the carboxyl terminus of each chain. It is important to understand that deoxyhemoglobin is related to the T state of hemoglobin. On the other hand, the oxyhemoglobin is related to the R state of the hemoglobin. Okay, so now how oxygen cooperatively binds to hemoglobin continued. It is important to remember that the R state is more biochemically active than the T state. There is a structural transformation that occurs when oxygen binds to the heme structure. This structural transformation ultimately moves the overall structure from the T state of hemoglobin to the R state of hemoglobin. More specifically, the moving of the iron ion encourages the movement of the histidine residue that is attached to the 5th coordination site. In addition, the histidine residue is a component of the alpha helix. This ultimately means that the alpha helix in the structure will move. In the alpha helix, the carboxyl terminal end interacts with the alpha-beta dimers. All of these factors contribute to the changes in the quaternary structure of hemoglobin, which also contributes to the shift of the T to the R state. The 2 pairs of alpha-beta dimers will rotate approximately 15 degrees. And now, the subunits of the structure have the ability to communicate more efficiently to the structural changes that have occurred as a result of the oxygen binding to the heme. So behind my face right here, you can see the quaternary structure how it changes and the rotation and everything I just previously mentioned. Okay, so how oxygen cooperatively binds to hemoglobin continued. Hemoglobin requires a combined model when it comes to cooperative binding. A concerted model needs to be mentioned because when hemoglobin has 3 oxygens attached to it, it is usually in the quaternary structure that corresponds to the R state. It is important to understand that in this model, the last open binding site is 20 times more likely to have an oxygen bind to it in comparison to a deoxygenated hemoglobin, which has no oxygens attached to it. On the other hand, a hemoglobin that only has 1 oxygen attached to it ultimately has the quaternary structure in the T state, which is the sequential model. In comparison to the hemoglobin molecule with no oxygen attached to it, a hemoglobin molecule that has 1 oxygen attached to it is 3 times more likely to attach further oxygens to it. Therefore, this explains why the combined model needs to be used in order to describe the cooperative binding of hemoglobin. Okay, so moving forward, binding capacity of oxygen to heme in the lungs versus in the capillaries. It's important to understand the difference in the binding capacity of oxygen to heme in the lungs versus into the capillaries. When talking about the lungs, the oxygen binding is saturated when approximately 98% of the binding sites of hemoglobin are taken up, which you can see on the graph to the right. So then in comparison, moving away from the lungs and into the capillaries and the tissues of the body, the oxygen binding is saturated approximately 32% of the binding sites on the hemoglobin are taking up. Therefore, the binding capacity of oxygen to heme in the lungs is much higher in comparison to the capillaries and tissues of the body. Okay, so how CO2 interacting with water can affect the pH of the capillaries. It is important to understand how CO2 interacting with water can affect the pH of the capillaries. First, CO2 enters the tissues and then into the red blood cells, which you can see into the tissues and into the red blood cells. Once it's in the red blood cells, the CO2 associates with water, which ultimately generates carbonic acid. This reaction is sped up by the use of carbonic anhydrase. From there, the carbonic acid breaks apart to generate HCO3- and H+. As a result, in the red blood cell, the pH would drop, which would ultimately make it more acidic. Okay, so that is the conclusion of this presentation, and that was the citation I used, and thank you.